Hello, Pima Aztecs. Welcome to Chapter 15, the Special Senses. So this will be one video on equilibrium. And here's the cartoon associated with equilibrium and all the special sense receptors it takes to maintain equilibrium. So we've seen this before in the previous hearing video. This is the vestibular apparatus. So this is all the vestibular apparatus. This all works for equilibrium. This guy, the cochlea, that's what we did for hearing. So the vestibular apparatus here is for equilibrium and orientation of your body. So let's take a look at the top. So we've got this cartoon. It shows the outer, here's the middle, and then here's the inner ear, and then the inner ear is enlarged. And this is this bony stuff out here, that's the temporal bone, which makes sense. Temporal bone, that's where your ears are located, over the top of the temporal bone. And this shows the semicircular ducts. That's these blue things. These are the ducts within the semicircular canals, which are just the carved out portions of the temporal bone that the ducts can run in. And then you have an anterior duct, you have a posterior duct back here, and then you have a lateral duct here. And then where these ducts kind of flare open, that's called an ampulla. So the area is the ampulla. And within the ampulla of the canals, or the ducts and canals, I guess, are these receptors called the crista or cristae ampullaris. And then another part of the vestibular apparatus is this area. So just outside of those just outside of the ampulla of the semicircular ducts and canals, you have this whole area which is called the vestibule. And then you have these spots called macula, these purple spots within the vestibule. And those macula are associated with the utricle and the saculae. So the vestibular apparatus then consists of equilibrium receptors in the ampulla. So we talked about those. There's 1, 2, 3 equilibrium receptors in the ampulla of the semicircular ducts in the canals. And then there's the utricle and the saculae of the vestibule. So the vestibular receptors monitor static equilibrium. So static just means kind of staying the same, and equilibration or equilibrium kind of means balance. Okay, so these vestibular receptors, the utricle in the vestibule and the saculae in the vestibule, those are the vestibular receptors, and they monitor static equilibrium. These receptors in the ampulla, the semicircular canals, they monitor rotational equilibrium, and we'll talk about that later. Oh, no, we won't. It's funny. Well, we will talk about it in more detail later. But yeah, so like I just said, these receptors in the ampulla of the semicircular canals, they monitor rotational and dynamic equilibrium. So dynamic means active movement equilibrium. Static means kind of staying the same equilibrium. I don't think the names for these are very descriptive, but that's what they're called. So now we're in the vestibule. So this slide we're talking about in the vestibule. So we're here. So this is all vestibule. Vestibule just means, I think it's Latin for opening. So you have these sensory receptors in the vestibule for static equilibrium and linear acceleration. And those receptors actually live in the maculae of the utricle and the saculae. These receptors basically monitor the position of your head in space, and they're necessary to control your nice upright posture. And we're not going to go into like the physics and detail of static equilibrium and linear acceleration, but just from the names, I think you kind of know that if these sensory receptors in the utricle and the saculae are there, they're helping monitoring where your head is in space, so that you know how to control your posture. So these guys respond to linear acceleration, but not rotation. So basically all that means is linear acceleration would be like if you were sitting in your chair and all of a sudden you stood straight up. That would be like vertical linear acceleration. And if you're standing up and then you'd sit back down, that would be linear acceleration. Rotational acceleration, that's if you spun around in your chair. So without going into too much detail, that's kind of the difference between linear acceleration and not rotational acceleration. Okay, enough physics. Now let's get into the fun part, the cells. So here we are in the vestibule. So remember, these guys out here, these are all the canals, and this is the vestibule of the, well, the the vestibule of the equilibrium apparatus. And then when they first saw these, the scientists called this a spot. And a spot in Latin is macula, or for singular maculae. So these are the macula of the vestibule. And then because they have different orientations, or they're I don't know, I kind of circled it, maybe you can't see, but the macula of the utricle is built like this, and the macula of the saculae is built like this. It's vertical, the utricle is horizontal. And that's going to help you respond to different forces acting on your head. So let's take a look inside one of these. This is actually looking at the macula of the utricle. So we've got our old friends, the cells, and they're hair cells. Just like if you listen or if you read about hearing, you know that we had hair cells with stereocilia in them for hearing. Well, Mother Nature decided to use hair cells. So here's a hair cell. Here's its stereocilia, so just like in hearing. But then in equilibrium and orientation, you also have this other thing projecting out of the hair cell. This guy right here called it kinocilium. Kinocilium. And you have this kinocilium, the top of that thing is embedded, well, with the hair cells, with the stereocilia, I should say. The kinocilium and the stereocilia, the stereocilia of the hair cells is embedded in this thing called an autolytic membrane. And it's kind of like, well, I've never seen it, but it's been described as kind of like Jello, Jello. So it's kind of like Jello, and then to kind of weigh it down, you have these crystals called otoliths. I'm not sure, I think I have it on the slide. I think they're like calcium carbonate or something. Anyway, they're autolytic crystals, or otoliths that help weigh the autolytic membrane down. And then there's also these support cells that help support the hair cells. And then the vestibular nerve fibers are here. So here would be the parts of the vestibular nerve wrapped around the base of the hair cell. So if you kind of understood how hearing works, you can understand that when you move your head in different, well, in like a linear acceleration, or if you move forward, that's linear too. What's going to happen is the otolithic membrane is going to move as you move forward, or in a linear fashion, I should say. And then that's going to cause this kinocilium to bend. And then you'll basically get a whole bunch of cool biochemistry, and graded potential will be turned into an action potential in the nerve fiber or axon of the vestibular nerve. Okay. Yeah, calcium carbonate stones. All of these components, so the kinocilium, stereocilia, otoliths, the membrane, they're all essential in static equilibrium and linear acceleration. So like I said, without going into the physics of what those are, you should know that the macula of the utricle and the macula of the saculae are involved in static equilibrium and linear acceleration. So let's look at the orientation of the maculae within the utricle and saculae. So here's how the utricle utricle is oriented. So here's the yellow. Remember, that's the vestibular nerve, and the hair cells are here with their stereocilia and their kinocilium. Here's the otolithic membrane. So, and then here's the saculae. It's oriented vertically. So here's the hair cells with their stereocilia and the kinocilium embedded in the otolithic membrane here. And then here's the fibers of the vestibular nerve. So you can imagine if you move this way or to the left, right or to the left, this membrane is going to move. And then, so if you move this way, the membrane is going to slosh back that way, and that's going to activate the kinocilium. Same thing with the saculae. If you stand up, or from a standing position you sit down, that's going to move the otolithic membrane, and then that's going to tug on the kinocilium, and they'll have a bunch of cool biochemistry to activate this vestibular nerve to tell your brain your equilibrium and orientation of your body. Okay, so the macula is horizontal in the utricle. We said this. It responds to horizontal linear acceleration. So that means if you're going forward or backwards, and then also tilting your head side to side, the utricle will respond to that. The macula is vertical in the saculae. That's what we said here. This is a vertical orientation, and it responds to vertical linear acceleration, like I said, standing up or sitting down. So this is a cartoon talking about activating maculae receptors. So here's our hero, the otolithic membrane, and the otoliths, those crystals. Here's our hair cell with its stereocilia. Here's the kinocilium. So this is all embedded. The stereocilia and kinocilium are embedded in the otolithic membrane. And then it says here what it's telling you in this part of the cartoon, of receptor potential. So remember, well, maybe don't remember, but a receptor potential is a graded potential, and that means it depolarizes the cell. So this hair cell, upon activation by the kinocilium and the stereocilia, it will depolarize from maybe -70 to maybe -60, so it'll be a slight depolarization. But if there's enough depolarization in this hair cell to excite this vestibular nerve, then you'll get an action potential here in the axon. You're getting action potential, and your brain will know that you have, well, in this cartoon, tilted your head. Okay. All right. And then, so these little hash marks, I don't know if you remember those. If you see these hash marks along an x-axis in the nervous system, those are action potential. This is a frequency of the action potential. And then we have depolarization of the hair cell membrane and hyperpolarization of the hair cell membrane. So the nerve impulses through a series of biochemical steps that we're not going to go through. You have, just as a baseline, you've got nerve impulses are generated in the vestibular nerve when your head is like this. So you haven't tilted it either way. You got like a baseline low frequency of action potentials generated in the vestibular nerve. Now you tilt your head, so here, second head. When the hair cells bend toward the kinocilium, so the kinocilium would be right here. When the hairs bend toward the kinocilium, then the hair cell will depolarize, and then that will excite, so that the hair cell depolarizes in a greater potential, and then that's going to excite this nerve fiber, which is going to generate even more frequent action potentials than baseline. So you can see these hash marks. That's showing you a higher frequency of action potentials versus baseline. And then in this one, this cartoon, you tilt your head the other way. Now the hairs bend away from the kinocilium. Actually, the cell's going to hyperpolarize. That's going to ensure that you have an even lower frequency of action potentials from the baseline. And you're going to inhibit the nerve fiber. That's going to decrease the frequency of action potential in the vestibular nerve. Okay. Yeah, so it doesn't go into a lot of detail of what happens, like all the biochemistry and cell physiology that happens within the hair cell, but it just says that the head position moves your autolytic membrane. That's going to tug on the kinocilium in the hair cells. When you bend the stereocilia in the direction of the kinocilia, you're going to depolarize your hair cells. So depolarizing, they're going to get more positive. That's going to increase frequency of action potential. So that's what it shows here. Depolarization increases action potentials. And if you bend in the opposite direction, you're going to get a decrease. You're going to get a hyperpolarization and a decrease in frequency of the action potentials. But this is the mechanism that Mother Nature designed so that your brain is informed of how your head is changing position. Okay, so we just finished this part, the vestibule, with the utricle and saculae. Now we're going to look at the semicircular canals and the crista ampullaris, so these little purple guys here. And then if they enlarge that, so this would be the semicircular canal. This is the ampulla. See how it flares out? So the ampulla flares out, and then within that ampulla you have this apparatus for dynamic or rotational equilibrium. So that, for example, if you just spun around in your chair, would be rotational equilibrium. But it's kind of designed the same way as the equilibrium apparatus in your vestibule, but it's a little different. So you've got the hair cell, which is typical. You have a hair cell here. Sorry, here's the hair cell. It's got stereocilia and a kinocilium, just like in the vestibule. And then it's got this thing over the top of it. It's not an autolytic membrane, but it's called a cupula. It's kind of the same thing. It's sort of like a gelatinous mass that the hair cell, stereocilia, and kinocilium are embedded in. There's one receptor in the ampulla of each semicircular canal. So remember, there's anterior, posterior, and lateral semicircular canals. The major stimuli for these guys, these hair cells, to get excited and stimulate, here's the vestibular nerve fibers, and excite those nerve fibers are rotary movements. So that's rotational. movements. Okay, said every crista ampullaris, it's got, so we talked about this, it's got the hair cells, stereocilia and kinocilium extend into this gelatin cupula. And then here's the dendrites of the vestibular nerve wrap around the base of the hair cells. And then, so when you're spinning around, this cupula is going to move. It's going to excite these hair cells. Those hair cells are going to send a signal to the vestibular nerve fibers, and you'll get an action potential in those fibers / axons, and that information will be sent to the brain to tell you that you are spinning around like a lovely ice skater. She looks kind of like from the 1950s or something. But anyway, here's her ice skates, and here she's spinning this way. And then it shows when she's not spinning. So here's the ampulla. Here's the little hair cells with their stereocilia and kinocilium. Here's the gelatin cupula. Here's the fibers of the vestibular nerve. You can see that cupula is not moving. When she starts spinning one way, the cupula will move. That will excite those hair cells, and they'll get that information to the vestibular nerve. So these cristae, or crista ampullaris receptors, so this whole thing is a crista ampullaris receptor. Collectively, they're called cristae. A cristae responds to change in velocity of the rotary or rotational acceleration of the head. And some details: when you rotate your head, there's endolymph in the semicircular canals. So I don't know if you remember endolymph and paralymph from the hearing apparatus over here in the cochlea, but there's endolymph in the semicircular canals, and when that endolymph moves, because you're spinning around, that's going to bend this gelatinous mass called the cupula, and that's going to pull on the hair cells. And that triggers action potentials to the brain through the vestibular nerve. And this gives the details. Here's the cartoon fully filled out, and then this is cool. This is a scanning electron micrograph of a crista ampullaris, and here's the cupula. So this piece out here would be the ampulla, 200 times medication. This is a cartoon of the equilibrium pathway to the brain. So it says at the top, input. Information about the body's position in space comes from 3 main sources: the vestibular receptors, the visual receptors, and then those somatic receptors or proprioceptors in the muscle and joints, and then somatic receptors in the skin. All those 3 entities give your brain information about where your body is in space. And then all that information is fed into 2 major processing areas in the central nervous system. First major processing area is the cerebellum, and then the vestibular nuclei in the brain stem. So for example, you can see information going into cerebellum comes from your vestibular receptors. Information about where your body is in space comes from visual receptors, and information about where your body is in space also comes from your proprioceptors in your muscles and joints, and then other receptors in the skin. And then the vestibular nuclei in the brainstem can communicate with the cerebellum, but the output of all this input information from these receptors goes to the oculomotor control and spinal motor control nerves, and then you can get this reflexive control of the muscles serving the eye, neck, limb, and trunk. So where should I look? Where should I move my head? That would be the output, or the reflex that would come out of synthesizing information from the vestibular receptors, visual receptors, and somatic receptors. And then I don't know if anyone's ever had motion sickness, but that results from conflicting information and confusion between two of these receptors or all three of these receptors. And there's conflicting information, you can get motion sickness.